My name's Mark Dawson. I'm a clinician scientist and I currently work at the Peter MacCallum Cancer Centre in East Melbourne. I did medicine in Melbourne and trained at St Vincent's Hospital. I did my basic physician training at St Vincent's Hospital and my specialty training on the other side of the Yarra at the Alfred. And after this time I really wanted to learn a little bit more about exactly how things worked in the body. And so I really committed myself to doing a basic science degree then. And I took that opportunity to go to the University of Cambridge to do my PhD there. And during my PhD I really got immersed in the field of epigenetics. What is epigenetics, you might want to know. So epigenetics has many definitions. We have great examples of this in nature, for instance. One of the best cited examples of this in nature is the caterpillar and the butterfly. Essentially, at the DNA sequence, these are identical. But you and I know that they're very, very different looking animals. And so the way our DNA is expressed at the right place at the right time, well, this by and large sort of encompasses the study of epigenetics. And as you can imagine, this is incredibly important for normal development of our various organs and tissues. But also, if this process gets corrupted, you can also see how very clearly this corruption of this epigenetic process that coordinates the way our DNA is expressed can also lead to cancer. And I guess that's one of the main things we've learned over the last 5 to 10 years, is that central to the development, the initiation and maintenance of several cancers, especially blood cancers, is abnormal epigenetic regulation. And that's really what I spend a lot of my time now studying. So since coming back from the University of Cambridge, we spent just over 7 years there and learnt a great deal of very important skills that we've managed to bring back and start our own laboratory here. Our remit has largely been to try and understand how blood cancers develop, how this abnormal epigenetic regulation contributes to the development of these cancers, and then try and correct this process using various drugs. I still do clinical work. I spend time seeing patients in the clinic here, and I think that really informs us about how important the task that we're doing in the laboratory really is. It's a difficult career, being a clinician scientist, because you have to really excel in two different but very disparate fields. The study of basic science as well as clinical medicine. At least in me, they extract two very separate sides of my personality. I could never see myself not seeing, speaking and treating patients. I get a great deal of enjoyment out of the time I spend with them. And in many ways it's also very rewarding and instantly rewarding. A smile, a thank you. These are the things that as clinicians we get every day. The other side of my personality is perseverance. And that's really what you need in the laboratory, because there's no gratitude in the laboratory. Things largely don't work, and there's a lot of trial and error, and it takes years to make incremental benefits count. But I think by coupling these two, we're really on a path to try and change the natural history of some of our aggressive, incurable cancers. One of the things we're really excited about is our upcoming move to Parkville. I think when Peter MacCallum moves to the Parkville campus and becomes integrated into the Victorian Comprehensive Cancer Centre, I think this will bring with it a real opportunity to interact with a critical mass of scientists that have specialties in other disciplines. Science by and large is now a multidisciplinary approach. Even though our central question is understanding how blood cancers develop and how we can better diagnose or treat these cancers, none of this would be possible without the skills that people bring from other disciplines. This kind of convergent science we see in our everyday practice. For instance, we have mathematicians who work with us, and mathematicians are critical to this process because the analysis of some of our very big data sets, analysis of whole genomes, of whole epigenomes, of whole proteomes, all of this requires very special skills that by and large people trained in mathematics are now able to apply to biology. Similarly, one of our greatest ambitions is to generate small molecules that change the natural history of some of these aggressive cancers. This cannot be done in isolation and without the help and the convergent knowledge of the chemists that we work closely with. Medicinal chemistry has provided us with an opportunity to really look at what we previously thought we were not able to actually attract or target with small molecules. And we've really changed that. We've changed that on its head. In fact, a good example of what we've been able to take from concept all the way to therapy is a small molecule that affects the way two proteins interact right at the surface of the DNA. This is called an epigenetic inhibitor. And in Cambridge, with the work of some exceptional medicinal chemists who worked as part of a pharmaceutical company, we were able to not only develop this small molecule, but show its actual preclinical application in the laboratory, in various assays that we have in the laboratory, and in also animal models of leukemia. And based on this really strong collection of data, we've been able to take the small molecule, not just from the bench, but all the way into the clinic. And I guess this is exactly why I am a clinician researcher, because not only am I able to see through some of these discoveries at the bench, but I have this great opportunity to actually take this into the clinical arena, and that's exactly what we're doing with this drug. So this drug is going to be, for a clinical trial with this drug is going to commence at the Peter MacCallum Cancer Centre in early 2015, and we'll be one of only 5 centres around the world that has the opportunity to give this drug to our patients who have otherwise exhausted all the other conventional therapies that we currently have. None of this great vision would be possible without, the real intellectual input that we get from other disciplines to feed into biology and ultimately to change our clinical practice.